All right, so good morning, everyone. So today I'm going to be sharing with you some data that we collected last year involving the use of static magnetic stimulation and its effects on axonal plasticity and cortical neurons. So static magnetic stimulation is a form of non-invasive brain stimulation that uses a rare earth neodymium magnet that has a constantly non-changing static magnetic field. Basically, you can put this up to your head and these static magnetic fields pass through the scalp to affect the underlying brain tissue. And as you can imagine, this is a very cheap and convenient form of brain stimulation. You don't require any specialized equipment. It's essentially just a magnet placed to the scalp. And it's typically seen to inhibit neuronal activity and reduce cortical excitability in underlying brain tissue. But the mechanisms underlying these effects are still relatively unknown. And in our lab, we specifically look at the effects of neuromodulatory interventions on axon plasticity. And so we wanted to explore, does this form of non-invasive brain stimulation actually change axon plasticity in some way? And when we talk about axon plasticity in our lab, we specifically focus on plasticity of the axon initial segment. And the axon initial segment, if we just take this kind of simple diagram of the neuron here, is the proximal part of the axon that actually generates the action potentials for neuron communication. And it's characterized by a very high density of sodium channels that allows it to depolarize and generate these signals. And so because it generates these signals, it's a very potent regulator of neuron excitability, basically deciding will a neuron respond to something or not. And what's really interesting is that this axon initial segment is plastic. And when we say it's plastic, we mean that it actually changes its morphological structure in response to brain activity. So what do I mean by this? So let's just say we've got that typical neuron example here with the axon initial segment. In periods of reduced activity, we see that the axon initial segment of individual neurons will actually lengthen, or they will relocate to move closer to the cell body. Now, both of these two changes result in increased neuronal excitability. And we see the opposite effect happen. So when there's periods of increased activity, the axon initial segment of these neurons will do the opposite effect, where their AIS will shorten, or it will distally relocate to move further away from the cell body to decrease neuron excitability. And we think that this kind of plasticity is a homeostatic response to activity to basically ensure so the neuron can fine-tune neural excitability so that it's always operating within these kind of optimal parameters. And again, because static magnetic stimulation has been seen to modulate brain activity, we then wanted to explore, can this form of stimulation actually induce plasticity of the axon initial segment? And so that's what we aim to demonstrate in cortical neurons in vitro. And we hypothesized because static magnetic stimulation has been seen to inhibit brain activity, then maybe we will see these changes that I've demonstrated here. So either AIS lengthening or moving closer to the cell body. So we opted to do this in culture. So we obtained the brains of postnatal day 1 C57 black 6J mouse pups. We dissociated the cortical neurons and seeded them into cover slips. And we maintained these in culture for 7 days. And then on the 7th day, we changed the media and the cells were stimulated. And the stimulation groups that we had for this experiment was a control group where there was no intervention applied to the culture well of the media. Our magnetic stimulation group, and this was static magnetic stimulation was delivered with a small 13 millimeter 0.5 Tesla neodymium magnet, which was placed underneath the culture well or the cover slip, so the magnetic field was always in contact with the neurons growing on the cover slip. And then we also had an additional stimulation group of 15 millimolar potassium chloride dissolved into the culture media. This is known to depolarize neurons in culture, and it has been seen to induce changes in AIS plasticity, typically seen to reduce neuron excitability. So this was our positive control. And we looked at this at different time points. So 6 hours of stimulation to assess the acute effects, 24 hours after the 6 hours of stimulation to see if there's any long-lasting or delayed effects. And then also an additional group where there was 48 hours of constant stimulation to see if there was any dose-dependent effects. And then these cells were fixed. We did immunofluorescent imaging and then quantified the AIS length and distance from the Soma. So we immunolabeled with these neurons with a variety of markers. One was MAP2 to visualize neurons, dendrites, and Soma, which are seen in red. We stained for the axon initial segment by labeling for anchorin G, the main scaffolding protein of the AIS, which is seen in green. And then we also labeled for inhibitory interneurons with GAD6567. So the data I'm showing you is from inhibitory interneurons. And from these images, we used a custom MATLAB script to quantify AIS length. And then we manually measure the distance that the AIS begins from the Soma in Fiji. And then we used non-parametric Kruskal-Wallis tests with post-hoc comparisons to see if there's any differences between groups. So now onto results, specifically looking at the length of the AIS first. We found that when we stimulated these neurons with potassium chloride in all our conditions, whether that was 6 hours, 24 hours after the 6-hour period, or for 48 hours, the axon initial segment significantly reduced in length when compared to control neurons. Again, this has been seen before and it's typically seen to reduce neuron excitability. And with static magnetic stimulation, unfortunately, we didn't see any significant differences in axon initial segment length at any of our time points when compared to control neurons, demonstrating that static magnetic stimulation doesn't seem to induce changes in the length of the AIS. And when looking at the distance from the Soma, we found that we didn't see any changes in AIS length with 6 hours or 24 hours after the 6-hour stimulation, but we did see a distal relocation at 48 hours of stimulation. Again, this kind of change has been seen before and is typically seen to, again, reduce excitability. And with static magnetic stimulation, we didn't see any effects with 6 hours of stimulation or 20 hours later after the 6-hour stimulation. What was really interesting was that when we gave 48 hours of constant stimulation, we saw that there was a distal relocation in the axon initial segment of these static magnetic stimulated neurons when compared to their control counterparts. And this was very similar to potassium chloride, which was very interesting. So all in all, we wanted to demonstrate, does static magnetic stimulation induce AIS plasticity in cortical neurons? And yes, it did. And this is really exciting because this is the first evidence that this form of brain stimulation can actually induce morphological changes in the axon initial segment. Specifically, what we saw was that the axon initial segment distally relocated, so it was located further along down the axon. And this only happened with 48 hours of stimulation. So it seems to suggest that there might be a possible dose-dependent effect of static magnetic stimulation. So the longer you apply for, the more likely you are to see an effect. But what was really interesting and quite puzzling at first was that the change that we saw, the distal relocation, was very similar to potassium chloride. And potassium chloride is known to depolarize neurons. So we saw a change that's typically seen to reduce excitability. So this then begs the question, what is static magnetic stimulation doing in culture? Is it also possibly increasing activity? And that's why you see this kind of change that's more reminiscent of reducing excitability. And currently we don't know. And so we're further investigating the cellular mechanisms underlying this process, specifically looking at patch clamp experiments to see if the morphological changes actually reflect excitability changes in these neurons. And then also, what is the mechanism behind this? Is it voltage sodium channel driven, or is it calcium channel driven? But this is very preliminary data, but it's very interesting, and it may suggest that static magnetic stimulation may be a promising tool to target and induce axon plasticity. And with that, I'd like to thank my supervisor, Dr. Alex Tang, and also Miss Emily King, who's talking after me, talking about her studies involving this other forms of brain stimulation. Thank you very much.